Jason Ernst. I'm faculty here at UCLA, and I'm going to be speaking about computational approaches for understanding and encoding human genome. So what you see in front of you is a small portion of the human genome, and it's with over 98% of the approximately 3 billion bases of the human genome. None of these nucleotides code for proteins. And there's a lot of interest in this sequence, in part driven by one of the results that came out from genome-wide association studies, that the vast majority of genetic variation associated with common disease is falling into these non-coding parts of the gene. So the sort of question for the field is, how do we sort of make sense of variance and, in general, the sequence that doesn't code for proteins? And in particular, we're going to primarily focus on potential regulatory. So we're going to, in this talk today, sort of overview 3 different high-level strategies and specific methods within them. One is using epigenomic-based signatures. Another is high-throughput functional testing data. And the third one is using comparative genomics signatures. So the idea with epigenomic signatures is that oftentimes reading the DNA sequence directly is very difficult. But if there's markers on top of the DNA, more important pieces of the DNA, that could be a strong signal that there's something important at that location in the genome. So it might be easier to recognize these different markers that are on top of the DNA. You think of it as post-it notes in some book. And biologically, we have these types of markers, forms of different types of chromatin marks, and in particular, we'll talk a lot about histone modifications. So these are modifications to the amino acids of the nucleostome proteins for which the DNA is wrapped around. And there's different types of chemical modifications in terms of methyls and methylation, and also specificity in terms of the number of occurrences in which specific amino acid residues are modified. And one important aspect of this type of data is there's the combinatorial patterns of these marks, which can give you a signal of important patterns from this data. In addition to the combinatorial patterns, there can also be spatial patterns, like broad domains of different marks. So we ideally would want computational methods to be able to recognize these types of patterns. And in addition, an important aspect of this type of data is also that, like the DNA sequence, which is virtually identical in different cell types of your body, as you go into different cell types, even within the same individual, you can have drastically different epigenomic modifications at specific positions. Oftentimes the marks, or a combination of marks, will be the same, but where they are across the genome can vary depending if, for example, you're in a blood cell, a T cell, or an embryonic stem cell. So what brings a lot of the computational opportunities and challenges is the ability to map genome-wide location of these different chromatin marks. So there's an assay called ChIP-seq, chromatin immunoprecipitation, followed by high-fidelity sequencing, which can map across the genome where different chemical modifications are. So a single experiment gives you tracks like this, which has sort of a density map where different marks are. So you're getting on the order of 20 million sequencing reads that are used to form one of these tracks. So the higher the value, sort of the greater presence of this modification relative to other positions across the genome. And you can see different marks that some have more of a peaky shape, others can have sort of a broader domain. And one of sort of the important things to note is that if you're interested in annotating the underlying genome, you don't necessarily want to just focus on one mark at a time, which a lot of analysis do, but also reason about the combinatorial patterns at some position, also the spatial context. So in order to do this, I developed a method that starts to focus, instead of looking at chromatin marks, at what we characterize or call chromatin states. In particular, it's a method called ChromHMM, which uses multivariate hidden Markov. So what you're seeing here, the rows correspond to different states, and then the columns correspond to different marks. And then the darker the value means a higher probability of observing that mark in your next state. And then once you've learned one of these models, you can go across the genome and assign each location to be an instance of the state. And that's the bottom row. So you've now reduced all that information in these different tags to chromatin state encoding, which you could color code and look at in the browser. And then once you've assigned each location one of these states, you can then ask what can they enrich for compared to other things that's already known, and start giving a characterization of these different states into different types of states. Like these are different types of promoters and cancer regions, insulator, transcribed, repressed, heterochromatic, and repetitive elements, so forth. So what we've effectively done here is we've now taken all this data and we've given a de novo annotation to the entire genome into these different classes of states. And just to give a little more detail about how this works in the modeling approach, so what we're doing is we're assuming there's these observed underlying biological entities, whether it's in cancers or starts of genes or transcribed regions, that you don't have annotated MPRA. What you have is these 200 base pair intervals with binary presence or absence of all different modifications, whether you have enough evidence that that modification is present within a 200 base pair interval. And we call this binary presence or absence if the number of reads exceeds what would be expected at a significance threshold based on a Poisson background distribution. And each bin here represents 200 base pair intervals, which roughly corresponds to the size of a nucleosome and spacer region. And then the mission probabilities are a product of independent variables. So that means for each mark, when you're in a specific state, you have some probability of observing that mark in that state. And all these probabilities are learned. Then there's also transition probabilities, which capture the spatial dependency through different states. So through that transition structure, for example, you could learn that for a reason that in this location here, where you don't have any H3K3673 marks, you could still be in that state 5, while when you go over here, this region, you might actually transition to a different state, even though at both locations you didn't see any marks. And to give another view of what we're effectively able to do here, what you're seeing on the top is the same genomic location, and then the different rows below that, or it's in 4 different cell types, is the raw input data, a heat map of it. And then for each cell type, we're encoding that into a chromatin state call, that information, in a cell type specific manner. And then we can summarize all that information. So that's for 4 cell types, but now we're summarizing that information now for 9 cell types here, and you can go into any location you're interested in, quickly see what the chromatin state is in any of those different 9 cell types. So we've learned this model sort of in a concatenated manner, treating additional cell types as if they were additional chromosomes. So we get one common set of chromatin state definitions across all the different cell types, but we can learn these cell type-specific state annotation. So that was in 9 ENCODE cell types. I've worked with the Roadmap Epigenomics Consortium, which tried to systematically map a large amount of primary cell and tissue types with various different histone modifications. And what you're seeing here is a grid where blue means that a specific mark was observed in a specific cell type. So that means we have experimental data tracked like this, that mark was available in that cell type. So one thing which we did with this is we learned a chromatin state model based on the 5 marks which were available in all 127 of these reference epigenomes, which span a diverse range of sort of human biology: brain, blood, stem cells, digestive, so forth. And this is a 15-state chromatin model based on those 5 marks. So one thing which you might notice is that there's additional marks here that we're not including in the model because they weren't mapped in every one of these cell tissue types. So the question here is, how can we define chromatin states uniformly across all cell and tissue types using more than 5 of the chromatin marks, including information from these marks which weren't available in all the cell tissue types. So the strategy we ended up applying here was to first impute the data that was missing, and actually also we created an imputed version of the data which we also had experimentally mapped. So what you're seeing now here is this grid where red means we have imputed data, and then the pink means we have both observed and imputed data available. And what you're seeing up here is in blue is observed experimental data, and red is the imputed version where we're not using information from that specific experiment to form that imputation. And you can see it agrees quite well visually, and we did a large number of quantitative comparisons, which are detailed in the paper. So the general strategy, how are we able to impute some of these tracks? So the idea here is we're trying to create a full chip seq track for some mark which hasn't been mapped in a specific cell type, but we're assuming we have data for other marks in that specific cell type, and that mark we're interested in other cell and tissue types. So the strategy I ended up developing was to use a method that takes a regression-based approach. In particular, we use an ensemble of regression trees where we're training on other cell types in which the target mark is available. And then the set of features in this regression are 2 classes. One is other marks in that same cell type we have. So marks can be well correlated with each other, and that correlation structure can be relatively well preserved across cell types. So we use the value of those other marks at that position we're trying to predict and other positions in the neighborhood. And then the other class of features is that missing mark we're trying to predict is similar to cell types where it's available. So oftentimes you have some mark you're interested in, and it's not available in that specific cell type that you could find in closely related cell types you have it available. So we have features that sort of define what are the similar cell types, and then also what's the average value of that mark in those in more detail. So once we had those imputed tracks, we then learned chromatin state model uniformly across the 12 marks for which we were most confident in the imputation for, based on being available in all in the most cell and tissue types. And we also used the imputed version of the data which we had available experimentally to make it more uniform across cell and tissue types. And also the imputed data had some advantages compared to the observed data because effectively we found also de-noises the observed data, because oftentimes the experimental data can be based on just 1 or 2 potentially noisy experiments. But when you form this imputation, you're combining information from dozens of very closely related experiments. So there might be a slight bias, but you're substantially reducing the variance, which ends up giving you often higher quality tracks. And then what you're seeing here now is this chromatin state model based on 25 states learned on 12 marks, now annotating the human genome into these 25 states across the 127 reference genomes, which arguably is one of the richest, most descriptive annotations of human regulatory. So this framework of chromatin state modeling is quite sort of general and it's been applied in a range of applications. These are just 3 that collaborations I've been involved in, applying this in context of studying melanoma progression, induced pluripotent stem cell reprogramming, and the 3D organization of developing brain. These were collaborations with Mel Ray's lab at Anderson, the class lab at UCLA, and the National Lab. So that this work was showing how we could use these epigenomic signatures to identify potentially interesting candidate regulatory regions. So these enhancer regions in particular, which could be these distal regulatory regions that are responsible for turning genes on and off, but could be potentially far away from any known gene. And one question is, can we have additional evidence that those regions that we're predicting are potentially regulatory actually are? And then furthermore, could we sort of dissect these regions we're identifying at a couple hundred base pair resolution to much finer resolution to identify the activating and repressive nucleotides within them. So this work is leveraging a sort of class of technologies that do high-throughput functional testing. So there's experimental methods which can now in high throughput test lots of different types of DNA sequences. Sort of two major classes of them. One is a massively parallel reporter assay where you take sequences you're interested in testing and then you put them on a plasmid and then test them. There's another class of strategies based on CRISPR knockouts that include this talk here, I'll give you an overview of this massively parallel reporter assay strategy. So in the specific variant of it that we used, and this was done in collaboration with R.J. Mickelson, who was one of the groups that developed this around the same time, one can synthesize thousands, or now up to 245 base pair sequences of DNA, and then get a quantitative readout of the ability of that sequence to drive gene expression based on putting that sequence in front of a reporter gene and then putting a barcode downstream of the gene. And then you transfect it and you get a readout of how many counts for that sequence give you this quantitative readout. So the question is, how can we leverage this technology to identify a close to nucleotide resolution within thousands of regions? What are the activating and repressive bases, and not knowing precisely which 145 base pairs to test? So the challenge here is that we have this finite constraint on the number of sequences we can interrogate, and that's a limitation of what we can synthesize and then also reasonably transfect and get robust readouts on. So the strategy here, the more sequences you devote to interrogating any one location, the fewer locations you can test overall. So the strategy we ended up converging upon ends up being a relatively simple design strategy, but it's quite powerful if you think about it the right way. So what we do is we take, in this case, 295 base pair sequence, and we simply tile it with 31 145 base pair tiles that are moved in 5 base pair increments. We cover 295 base pairs in total. So in a single experiment, we can test close to 8,000 regions. We have coverage of 295 base pairs. And this is important because from the epigenomic data, we can often be confident at a 2 to 300 base pair resolution where the most likely regulatory site is. But it's hard to be confident about a specific 145 base pair. And then we also have information available to recover at high resolution activating and repressive bases. So this point is where some of the computational challenges go. So how do we go from this overlapping sequences to inferences of the high resolution activating and repressive? So to give you some intuition here, this is showing you the 295 base pairs of sequence and then the 145 base pair subsequence. Now suppose there was an important activating sequence right here at that location where the yellow plus is, that was sufficient to turn on gene expression. Then we would expect the subset of tiles that overlap those positions shown in yellow to show high activity, while the remaining tiles to show closer to basal activity. And then if there was an important activating sequence at this position, we would expect these subset of tiles to have higher expression. And if there's an important repressive sequence right here, we would expect these sequences to have below basal expression. So this was just to give some intuition. Now here are two actual experimental data and showing you the inferences of this the probabilistic model, SHARPR, that I'll talk about shortly. So what you're seeing here is the inferences of the relative regulatory activity score at each position along the 295 base pair. And blue and green are based on using two different types of promoters. You can think of it as sort of stringent replicates. And then black is the overall combined inference. And then, so the method's effectively making prediction. There's an activating site right here and a repressive site at this position. And then if we go and compare this to predictions of regulatory sites based on DNA sequence information and combined with other DNA split printing, this was based on this method Centipede from Jonathan Pritchard's group. It makes a prediction that there's a regulatory site at those two positions in red. So you can see that it lines up nicely with our peaks of activation and repression. But there's sort of two key advantages to this type of information that complements the sequence-based predictions of regulatory sites. Or three, I mean, so one is that you have information that differentiates activation from repression. Second advantage is that it's making these inferences independent of actual DNA sequence. You could potentially identify regulatory sequences that don't match well any regulatory motif pattern. And then also this is orthogonal, it's based on actual experiments. So how we actually go about making these inferences is we have a probabilistic model that makes certain additivity and normal assumptions. So the y variables correspond to the observations after certain transformations of each of the 31 reporter tiles. And then we have a latent hidden variables associated with each 5 base pair interval that span the 295 base pairs. And then we assume here that the y variables, so y1 is going to depend on the h variables that it overlaps, so h1 through h29, y2 will depend on h2 through h30, and so forth. And then we assume that the y's are normally distributed with their mean depending on the mean value of the incoming h variables that it overlaps, and then the variance set to the empirical variance of observed data, and then the h variables. We also have a normal prior now on, and it has a mean set to the empirical mean of observed data, and we have one free variance parameter which controls the overall smoothness. And in practice, we use two different variance parameters and take a more conservative estimate after standardization. And more details of some of that. So what you're seeing now is the overall, or the inference of activity at all sites that had this motif NRSF, which is a known repressive motif. So red is the places that have this motif predicted based on sequenced information. Blue are other regulatory motifs predicted in these regions. And you can see that there's a dip where that regulatory motif is in many cases, quite a strong dip. And again, this method's making this inference without any knowledge of the DNA sequence or of where the regulatory sites are. It lines up quite well there. And then we can also use this type of information to sort of better characterize these regulatory chromatin states that we were inferring. So in this case, what this is showing you is the average activity of sequences that were in different chromatin states based on this assay. And what it's saying here is from one of these chromatin state models that we had learned that states that we characterize as the active promoter or the active enhancer 1 and 5 had above average activity. And we're looking here specifically at regions that were open accessible DNA, but they're differing on what histone modifications and other associated chromatin marks are present, while other sites, like what we characterize as a weaker enhancer, had above average activity, but not quite as high. That's in state 8, while there's other states that had far below average activity. So it's saying there's additional information in these chromatin states beyond just is the DNA accessible. In the last part of the talk, I want to talk about an alternative strategy for understanding the genome, in particular the non-coding genome. That is using comparative genomics signatures. I'll focus on a method we recently developed for learning conservation. So one of the sort of the original annotations of the non-coding genome was using comparative genomics information. After sequencing the human genome, they started sequencing the mouse genome, and then the rat and dog genomes, and then additional genomes, and start comparing the DNA sequence in these multiple sequence alignments to identify what parts of the genome are constrained evolutionarily. And it turns out about estimates around 5% of the human genome is constrained, and the majority of that is in the non-coding parts of the genome. And this information was, so one forms these multi-species DNA sequence alignments and then summarizes that traditionally in methods that either represent scores of evolutionary constraint or make these binary element calls. And there's some advantages of this type of information. I mean, one nice thing is you're getting single nucleotide resolution information. And another thing is that it gives you an annotation that something is potentially important in some location of the genome, even if you don't have experimental data in the cell type which that location is active in. So if some locations only active in cell types for developmental stages for which you don't have experimental data, you might otherwise not think that location is important. And then there's been methods to associate different annotations with heritability enrichments, and this is stuff that Ogden has been speaking about, and work from Ben Neal and Alcus Price's group. Under their assumptions, they argued that constrained elements have the greatest enrichment for phenotypic heritability. So it's sort of signifying there's something important about these locations. But we were also noticing there's some limitations of representing the comparative genomics simply as a score binary element call. So this is showing you two locations in the genome browser which have high phylo-P and PhastCons scores and lots of PhastCons elements. So if you just have the browser, the summary tracks on, they'll look like similarly important locations. But then if you turn on the underlying alignment tracks, you can recognize they're fundamentally different types of locations. The one on the right has this robust alignment pattern through many vertebrate species. So green means that that position, there's an alignment there. So the one on the right is going all the way through fish, while the one on the left sort of has this odd alignment pattern where you have these distal vertebrates aligning, but not any closer mammals. And this is it's an extreme example, but it's actually not that unusual, and it's making a more general point that there's a lot of information in the underlying alignment tract that doesn't get conveyed when you try to reduce it to a single score binary element call. So a student in my group, Adriana, worked on this project where we were trying to apply a similar type of a modeling approach that we had developed for epigenomic data in the context of comparative genomics. Instead of thinking about this comparative genomics multi-species alignment just as a score or an element call, we wanted to characterize it into conservation states based on the combinatorial patterns and spatial patterns of which species are aligning to and matching a reference genome. So what we did here was we took a multi-species DNA sequence alignment and we worked with the 100-way vertebrate alignment from UCSC genome browser. We encoded it with, for each species, one of three possible observations. Either aligns and matches the human reference genome sequence, so that's represented with the check. It aligns but doesn't have a nucleotide matching the human reference genome, that would get the X, and then it doesn't align to the human reference genome. Either there's an indel or just no sequence alignment in the region, and that would get this dash observation. And then what we did was we effectively applied, a similar model to ChromHMM, but we now generalize this to be a multinomial random variable. So for each species, we have one of three possible observations, opposed to before we had one of two possible observations. And we also are doing this now at single nucleotide resolution, where before it was done at 200 base pair resolution. So this also raised scalability challenges. we handle that by, instead of with ChromHMM, we would go across the entire genome in every iteration of the Von-Welch or EM algorithm. Here we would now subsample different parts of the genome of each iteration. So what you're seeing now is an illustration of this modeling approach where you have the different the human DNA sequence, and then we're inferring these different hidden states based on these observations after our encoding for the different species, and then different states are associated with different probabilities of those three observations. So now this is showing you an actual data. So this is the application of this modeling approach to 100-way vertebrate alignment. And what you see on the left is the probability that a species aligns to the human reference. So that's the probability of the check or X observation, or 1 minus the indel gap observation. And on the right side is the probability of matching. So red corresponds to a greater probability, blue corresponds to a lower probability, and then the columns are ordered based on the major clades and their distance to human, and then the individual columns are different species. And what you're seeing here, the top row corresponds to state which aligns and matches through all the vertebrates, while the bottom one has this pattern of having high probability through these distal vertebrates, not these intermediate mammals. And what we're seeing now, sort of going back to that motivating example, those two locations, which both look like they have a lot of constraint, now separate into very different conservation states. Where the one on the right, a lot of those positions get assigned to that state 1 with high probability through all the species, while the one on the left, a lot of that location is getting assigned to the state 100, where you have high probability for these distal vertebrates and not these distal vertebrates. And if we look at where PhastCons elements are, so this is an annotation of constraint based on traditional phylogenetic modeling approaches, we can see that their elements are falling both into that state 100, and then also a lot of the states up top, which are more, which have associated with higher aligning and matching probabilities. So it's showing on a systematic manner that we can differentiate what would otherwise be considered one homogeneous annotation. And to show that that example that I was showing you is not an isolated example, 4% of all the PhastCons elements actually fall into that state 100 based on the vertebrate alignment. And then we can start, similar to what we have done with the chromatin states, once we have an annotation of each location to one of these states, we can compare them to other existing annotations. So one thing which came out of this, which might not necessarily be expected, was there were states that heavily enriched for the transcription start sites of protein coding genes. So just based on the combinatorial patterns of what species are aligning, we saw a state which had 30-fold enrichment for transcription start sites. And it wasn't the state 1 which was aligning and matching for all the species. The signature of transcription start sites was associated with some probability for many species, but not particularly high probability for any one species. And then what we're seeing here, so for the state 100 that I was talking about before, and also another state 82, they heavily enrich for transcription start sites of pseudogenes. So what could be driving part of this state 100 could be alignment artifacts associated with pseudogenes. And the state 82 is a similar pattern to 100, but weaker, where it has some probabilities for these distal vertebrates, not quite as high, and not lower probability for many mammals. And that one still has over 50-fold enrichment around. And then one of the points I made with the comparative genomics data is that it can also give you information at single nucleotide resolution, and this is illustrating that, how we can have these different states, and pick out different nucleotides of the codons. So this is showing you relative to protein coding gene exon starts in phase 0, where we have this 3-base period oscillation, where at the third position, states 3 and 54, which are associated with distal alignment, but not quite as distal matching, like 54 aligns through most species that only matches through a small number peak at that third base period. So this is another advantage of this type of information is you're differentiating aligning and not matching from not aligning at all, which a lot of models don't effectively convey. And then if you look upstream, you can see a signature associated with the splice site as well. And we can compare the enrichments of the conservation states to the chromatin states. So what you're seeing in the rows are those conservation states which were maximally enriched for at least one of the chromatin states of the 25-state model based on the roadmap data that I had talked about earlier from the imputed data, and then below it is the different chromatin states. And what we're seeing here is that some of the conservation states, like the state 28, which I said was associated with transcription start sites, heavily enriched with chromatin state associated with transcription start sites. The one that was maximally enriched for enhancers is the state 2, which aligns through almost all the vertebrates except for fish, while the one that aligns through all the vertebrates has the strongest enrichment for the transcribed states where the exons are. And then there's other states associated with more heterochromatic and recessive information. This is again sort of making the point that the information in this multi-species alignment is very multi-dimensional. It's not necessarily, if you're losing information, just think about it in a single variable. And then this is another analysis showing there's additional information in these annotations relative to constrained elements. In this case, we took PhastCons elements and we looked at their enrichment within non-exonic regions for fetal brain DNase 1 positions. So these are positions that are open chromatin and developing brains of accessible DNA. And this is one of the more epigenomic annotations that is most associated with constraint in general. But what we see here is that depending on which conservation state the PhastCons element is in, it can have very different enrichments, where those PhastCons elements that are in a lot of these states here, which don't have a particularly robust alignment pattern, have very limited enrichment for these fetal brain DNA sites, not orange. If we look in blue, that's bases that are not in a PhastCons element but are in one of these states. And we still see enrichment for some of these states. It's not as high as when in PhastCons elements. It's saying there's additional information from this phylogenetic modeling, but it's also showing that because these enrichments vary a lot, there's additional information within these conservation states. And we also did another comparison here when we took constrained elements and we split them into two halves based on which conservation states they are. So we defined a set of top 7 states here that I'm showing you based on their enrichment within DNase data within the non-exonic regions, similar to what I showed you before, but based on a larger set of DNase scenarios. And then the PhastCons elements in the other 93 states. This divided the PhastCons elements into two halves. And then we applied the heritability partitioning that I mentioned before, and what we see is very different enrichments for phenotypes for the majority of them, depending on which half set. It tends to be higher enrichment for the PhastCons basis of these 7 states. It's more robust. So to summarize, I presented to you a chromatin state modeling approach for annotating the non-coding genome with annotations available in cell tissue types. Then I also talked about one method in the class of high-throughput functional testing approaches for identifying at high resolution, activating repressive nucleotides within thousands of candidate regulatory regions, and then showed you how conservation states provide a complementary single nucleotide. And I'd like to acknowledge the number of people who've been involved.
do I need to reintroduce myself? It says everything there is to know on there. I want to say this is kind of an old project from almost 10 years ago. So in terms of the tools and some of the ideas, it will look quite obsolete. But I thought it's good to talk about it, especially as the last talk in this week. Everybody is tired because it's a very cool story with some really interesting aspects to it. Also, maybe like the young people might learn some stuff about what we did in the old days from the things I talked about. So the title is a genetic soap opera. I need to justify that I use this very pretentious term to describe this story. So let's just make sure that we understand what it means to be a soap opera. So if you're watching a soap opera, you expect to see some amazing and unexpected plot turns. You're expecting to have suspense, cliffhangers, and surprises. Very important to have treachery and lying. And one more thing that's obviously always required from a soap opera is to have romance. Now my story doesn't have romance, but why do they put romance into soap operas? One major reason is that they could have models playing the main role, so we are going to Okay. The other thing, some very cool sayings that are really coming to life when you hear stories like that. One really nice one that all of you probably know is that nothing in biology makes sense except in the light of evolution, so we should always be thinking about the evolution that shaped the biology that we're seeing. And the other one, no less famous, can help you. And anyone who's worked with doctors in particular knows they're always very paranoid on who's doing something behind their back and not telling them and lying to them. So we'll see that sometimes paranoids do it anyway. Okay, so I have some very low-level introduction that I'll just breeze right through. So we all know how human genetics work with the inheritance models of the autosomal and non-autosomal pieces of the genome. And we all know, but it's very important to keep in mind this idea that the two main forces that shape our genome are the mutations that happen and the recombinations that basically very different combinations of the mutations by creating haplotypes over generation. So somehow these two processes work. The third one, of course, not mentioned here, but very important, is selection. That some combinations of mutations might have positive or negative effect on the survival of the species. And so these three things together basically create the shape of the genome, the genomes that we see. When we talk about functional genetics, then the point is that we have these two separate pieces of information. We have the genotypes and some properties of the organisms that carry these genomes or genotypes, and we're looking for connections between them. But what we are really looking for, so there's two different things we might be looking for, and I'm sure we all know the difference, but let me just read them. One is we're looking for statistical associations between them, but we are really hoping to get at the mechanisms that connect them through biology, right? So for example, not only the statistical associations, but the actual genetic causes of disease. This is also, this was written years ago, but I think the situation hasn't changed much in the sense that there's lots and lots of studies of connections between genetics and disease. We have a lot of associations, a lot of accumulating knowledge, but our medical understanding of the implications of this lags far behind our ability to find statistical associations. Okay, this is how GWAS works. I'm sure we all know. We're looking for these genetic patterns. The simplest one is just a single mutation that are associated with the genotype. And we are hoping to eventually be able to we're hoping to eventually end up with the biological causation, but just finding an association is definitely not yet finding a biological connection. Okay, we are still in the intro, and so of course we understand that what the genome encodes are these proteins and connections between proteins and different functions of proteins, and so we expect that most of the connections we find will be related to the genes and the proteins that come out of them. For example, changing the structure of the protein, and therefore, so changing the sequence is changing the structure, and changing the structure is changing the function, and this is kind of the most basic chain of logic that we would expect. And when we find risk factors that are Mendelian in nature or very strong, even if they are not Mendelian, we can often associate them with exonic mutations, such as BRCA, for example. Or genetic diseases that we understand exactly their heritability, the way they are inherited. But typically, and since this was written, I think this become much more widely understood, that the weaker associations that we find in complex disease, for example in GWAS, are typically not gene-altering mutations, but they refer to more complex mechanisms such as regulation. So, okay. This is an introduction that basically I was talking about things that most of the people here probably know more than me about. So we might move now to things that hopefully the audience doesn't know a lot about. So, and I also don't. So it would be a good fit. So we'll move to talk about epidemiology a little bit. This is still background. So what we are interested in here is chronic kidney disease. And I'm sure we all know from general knowledge that kidney disease is very common, is very expensive, and is very damaging to people worldwide and in the US in particular. So as of around 2010, or this is actually around 2006, it was claimed that 15% of the US population suffer from chronic kidney disease. And chronic kidney disease is not only being on dialysis, it's also just having some reduced function of the kidney that requires medication, even though you're functioning regularly. So I guess 15% of the US population were on some sort of kidney medication as of 2006, and about 8% DM is type 2 diabetes mellitus, and about 2.5% suffered from chronic heart failure. And the very important thing to notice here are these overlaps. Obviously, diabetes is closely associated with kidney failure, although actually the vast majority of kidney failure cases are not related to diabetes. And the other very strong connection is between heart disease and kidney disease. It is well known that people with kidney disease are much more susceptible or have much higher prevalence of heart disease than the general population. But one thing that is extremely interesting and very easy to do in the US is when you compare the ethnic disparity in these diseases. So, for example, many of you don't know, and I didn't know either, that the milder forms of kidney disease are actually more common in European Americans than African Americans. The advanced stages are much more common in African Americans compared to European Americans. And one thing that's really interesting is that if we look at African Americans that have end-stage kidney disease, the prevalence of heart disease is much lower compared to European Americans with end-stage kidney disease. Right? So the conditional probability of heart disease, given you have severe kidney disease, is high in both populations, but is much lower in the African Americans. For this, I have some nice graph. So this is again the conditional probability, right? So the prevalence of heart disease within people with end-stage kidney disease. We see, for example, people that also have diabetes. It's almost 40% in males. In European American males, and about 20% in African American males. And people that don't have diabetes, it's even a bigger gap for males, and I mean similar but less extreme picture for females. Okay, so we have this epidemic that's American-wide, but it has different characteristics in European Americans and African Americans, and very distinct different behaviors. So, so this is where this approach that was very popular, I guess 10, 15 years ago, so admixture mapping is meant to deal with a situation where we have significant racial disparities in some disease, for example, end-stage kidney disease. We know that we have significant disparities both in the prevalence and in the epidemiology of this disease between European Americans and African Americans. And we have a population of recent admixed origin, and we definitely have that, because African Americans are in fact an admixture of African and European ancestry. And the point of admixture mapping is, in that case, we can identify which regions in the genome are basically responsible for the genetic basis of the differences in disease etiology or disease prevalence in these groups. And we can do that by studying the genomes of these admixed individuals, healthy and affected, and in fact we can do it by studying affected genomes only. So this is kind of cool. I'll show you some version of the admixture. So this is called admixture mapping, or mapping admixture by linkage. So here's the idea. We have a guest appearance by David Schiffman, who's not here anymore. So basically what we can do is we take a bunch of African Americans. This example is not the sort of the color scheme is not exactly good for African Americans, because in this example we have admixture with a little bit of orange and a lot of blue, and the orange population is responsible for the risk. So in this case we see that the sick individuals, the cases have in this region in the genome that's responsible for the elevated risk, the cases, most of them have orange, or all of them have at least one orange, whereas the controls don't have orange. And because there's actually a small percentage of orange in the genome, then you know, this is just expressing the fact that there's a small probability of an orange variant in every point in the genome, and this is expressing the fact that to get the disease, you probably need to have orange in that region of the genome. With African ancestry in African Americans, the picture is a bit different, because we know that the major source of the genome is actually African for African Americans and not European. But conceptually, it's similar. We can divide the genome into regions, or somehow estimate in every point in the genome what percentage of the ancestry is from one source versus the other, and look for excess of African ancestry in a region in the genome. And the point is that we could do this with cases only. How can we do it with cases only? We can actually compare every individual to themselves, estimate the overall genome-wide percent of African ancestry for each individual, and look for region in the genome where per individuals and accumulated across all individuals we have excess of African ancestry. So we could do this with cases only. We don't actually need controls. And there's one specific algorithm that does it. It's called admixture mapping. It's hitting all the keywords for methods that we can think about. It's a hidden Markov model to infer what percentage of the ancestry is African at every point in the genome. So we know that this is a continuous process, right? If I have African ancestry in one point in the genome, I'm highly likely to have African ancestry close by. Why? Because the stretches of ancestry in my genome are actually determined by the recombination, right? If it's recent admixture, then each chromosome might be might have been broken in 10 or 15 places by accumulating recombination since the admixture event. And so I would have like 10 or 15 pieces to each genome. Within each piece, I would have a fixed origin, European versus African, right? So basically, the recent admixture means that relatively long stretches of the genome would be from the same continental origin. And within the HMM, we have an MCMC, so that now relates to my talk from last week. And why did people really like admixture mapping circa 2008, 9, 10? So one important point is that requires much less data than GWAS to identify the disease loci, because we have a lot of power because we have long stretches in the genome. So this really allows us to infer with high power, with a relatively small number of samples, which regions in the genome are. So this is not fine mapping like GWAS, or not pinpointing like GWAS, but this is identifying regions in the genome. So that's the advantage of GWAS. We have more power with less data. The advantage over traditional linkage is that it does not require Mendelian inheritance. So it doesn't have to be the same mutation, right? It's more similar to the overall gene load test that people are doing now, because it's identifying regions that might be a gene or several genes wide. So this was, and one very important thing is that to know is that at that point in time, people were not very rich, were not basically doing sequencing. So sequencing things as a way of solving problems was much less relevant than it is today. So we were limited to doing SNP chips, and it would be much cheaper and more realistic to do, say, 1,000 SNPs across the genome, which is what you need for a mixture, for admixture mapping, than do 1 million. I mean, the cost would be disproportionate. So that's why admixture mapping was really popular. And this end-stage kidney disease difference between European Americans and African Americans was really a good candidate for this kind of study. So people went and did this study. And in 2008, in Nature Genetics, 2 independent studies using, supposedly independent, I mean, they were using different data for sure, but they reached exactly the same conclusion. And the conclusion is trying to find the region in the genome where the genetic basis of this difference lies, right? So, and they both found the same position in the genome, same region in the genome, original chromosome 22, and they also both had a suspected gene, supposedly independently, and the gene name was MYH9. So the conclusion of both of these studies, and I just want to introduce 2 of the stars of our talk, Jeff Kopp and Cheryl Winkler from the NIH. So these are the big shots in kidney disease in the NIH then, and I think possibly also today. And so they were the first and last author of one of the papers. The other paper, I think, was from John Hopkins. Okay, so both of these
dates on those 2 papers? I mean, how close were they in time? They're both
they appeared at the same time. I guess they were submitted almost simultaneously. I'm sure they knew about each other, and they submitted. I don't remember the history. Maybe they submitted together, or maybe they submitted separately in the journal. Said we have these 2 papers. We should give both of them a chance, or publish both of them at once. They actually appeared in the same issue of Nature. I mean, it's very redundant to have 2 of them, but that's what they did. Okay, it's also we can talk about the politics behind it. So why MYH9? So first of all, there was some known connections between MYH9 and kidney disease. So these people, when they found that the peak of the admixture mapping was very close, was in the region of MYH9, they were really happy because they knew the name. As kidney doctors, they knew the name of MYH9 from some other phenotypes, but that are related to the kidney. for example, this second point, the term mutations in MYH9 had been shown to cause familial clusters. So in linkage studies, they were actually able to assert a real connection between MYH9 and kidney disease. So obviously it's a good candidate. So these were kind of the biological reason, and the statistical reason was that, so we are saying that what we're doing is we are, by the HMM, by the hidden Markov model, in fact, we are inferring at every point in the genome how much of the percentage of the ancestry is African versus European. And this is a continuous function because the HMM is sort of continuously, smoothly changing. And we can look where in the genome is the biggest difference between sort of the genome-wide African ancestry and the local African ancestry. And the first group, the NIH group, actually got that the peak is right on MYH9. So that was actually where the highest peak in the genome. So they had one SNP in MYH9 in the panel, and that was the point where the highest signal was observed in the HMM. And they also, I'm not sure how to do it for time. These links lead to figures and tables from the paper. The other thing is they did tests like we do in GWAS for some genotype mutations in MYH9, and they found a strong statistical association between these mutations, and which, if it didn't happen, it would be very surprising given that the mold peak was on MYH9. So you could say that they confirmed the statistical association by doing genotyping of SNPs and getting a strong, very strong statistical signal about association between MYH9 and end-stage kidney disease in African Americans. Okay, so it's also nice that they looked in other genes within that region. So that region was centered at MYH9 for one paper. For the other, it was actually centered a bit away from MYH9. So they said, I mean, because the signal we find is not super local, we can look at 5 genes to the left and 5 genes to the right and select some SNPs from those, genotype them as well, and look at the strength of the statistical association. And basically, to some extent, they found that whatever statistical association existed is explained by LD between these mutations and between these SNPs and the MYH9 SNPs. So all the signal pointed to the fact that we should be concentrating on MYH9, right? And the people leading this area are people who really care about the medical understanding and medical implications. And so, and this is a very important phenotype with a very striking finding in an interesting gene. So basically, everybody in the world was working on trying to understand this connection further. This is over about a year and a half from the publication of the original paper. This is just a sample of hundreds of papers that appeared. For example, a focus on the MYH9 susceptibility gene. This is related to HIV-associated nephropathy. So people with HIV are much more likely to develop end-stage kidney disease, and people with HIV and this MYH type thing are like 100% probability or many tens of percent probability to develop end-stage kidney disease. And the point is that I became part of a group that was also working in this area at that time. So this was actually a group in Technion led by Karl Skoretsky, who's a nephrologist and a scientist, and Doron Behar, who's a well-known geneticist. So these are people from Technion, and I was working with them. And we also collaborated with these two people, Kopp and Winkler from the NIH. And what we were doing is we were looking for interesting things that we can say about MYH9 associated kidney disease, trying to have new statistical perspective or new biological perspective. So this specific paper is about a not very interesting fact that the same effect that we see in African American replicates in Hispanic Americans, and Hispanic Americans have a much lower but non-0% of African ancestry. So you can actually find the same mutations in MYH9 in lower prevalence, and see that they are actually associated with end-stage kidney disease in Hispanics as well as African Americans. Not very interesting and very expected. I should mention that we had our data from this second to last guy, Walter Wasser. So the cases are just a bunch of cases from one clinic in New York City, just from one dialysis clinic in New York City. He just brought his patients with him, and we had some crappy control set to go with it. And so we were collaborating with Kopp and Winkler in having these weekly or bi-weekly phone calls to talk about what interesting things we can do, both in terms of data analysis, statistical analysis, looking for new frontiers in this. And we're now in February 2010. April 2010, there was, to demonstrate how important this thing was, they had a special conference at the NIH about MYH9 in kidney disease, clinical and public health implications. And of course, this is an NIH. Two of the stars of this are our friend Kopp and Winkler, and we're talking about April 2010. Okay, so now we stand in April 2010. We're having conferences about MYH9 and kidney disease and what can we do about it. And what do we have as of April 2010? So we have many independent replications of these original statistical findings. For example, our Hispanics paper, which is just replicating the same thing in a slightly different context. So we know it's real. We know the statistical association is real. They did more and more fine mapping on MYH9 and found additional strongly statistically associated variants in MYH9. So we have one way of saying it, we have multiple independent signals that do not explain each other within MYH9. There's no progress on how the biological mechanism makes sense, and none of these mutations are actually mutations that are supposed to have a major functional effect. So I think they were all intronic, or might have been a couple of synonymous mutations in exonic regions of MYH9. So now we need to go back and ask ourselves, given that we have a strong statistical signal, but we really have no progress on what the biology is, should we really concentrate all our efforts on MYH9? And I was the one actually pushing it, that we should try to look for other genes and other mutations, and we had a phone call with the NIH people, and we told them maybe we should be looking in other places. That was the last phone call we had with them. We'll see why in a little while. But we didn't have good data. We didn't have a lot of money. And so the point was, let's look in the public databases. Let's look in the literature and find things that we think might be interesting. Then we can invest money in genotyping them and on the case control data and examining them further. So, for example, other mutations outside MYH9 that could conceivably explain the association within MYH9, given, for example, LD patterns. And the main source as of April 2010 was HapMap. I hope you all know what HapMap is. So HapMap was supposedly a map of human genetic diversity. Before we had sequences for different populations, this was supposed to be a pretty exhaustive map of the common variants in different populations in the world. So if you try to read and understand what HapMap claims to have, then it's supposed to have almost all common variants in different populations that have been studied. Okay, so for example, there's a large number of Yorubans, West African samples that are part of the sort of the original discovery samples of HapMap. So supposedly we would expect that most of the common variants in West Africa would also be present in HapMap. But my search of HapMap and other resources found no promising candidates. Yeah.
What was the
in MYH9? High, like 30-40% in say YRI in Africans, and maybe 10-15% in European. There was major frequency difference, of course, because we're looking for something that the allele responsible for it should have major frequency difference between the populations, but it was pretty common in Europeans as well. Okay? And this should be a hint, right? Or it was not super rare in the Europeans as well. This should be a hint because this, I haven't really talked about it, but one, and we didn't understand it until later, one major difference between end-stage kidney disease, non-diabetic end-stage kidney disease in African Americans and Africans against Europeans, is that you have non-negligible several percent or like 1% in the population prevalence of end-stage kidney disease that's very early onset in African Americans. Like people by age 40, they need a kidney transplant, and this basically has zero prevalence in Europeans and European Americans. And so this tells you, this should be a hint that we didn't really think all the way through then, that what you're looking for probably has zero prevalence in Europeans. Okay? So, so at that point, something that's well established today was only in its pilot stage, which is the 1,000 genome. So 1,000 genome is a way to not depend on HapMap for finding variants, to be able to find your own variants. So around April or March, the first pilot data of the, March, April 2010, the first pilot data of the 1,000 genome project came out, and it was 60 European genomes and 59 West African genomes. And once you start to look in there, you suddenly see completely different things. Okay, so we said let's focus on the most smoking gun that we can find, which is non-synonymous coding mutations that are in genes close to MYH9 and that they have major differences in allele frequency between Europeans and African. Basically, they have to be relatively common in Africans and missing completely or almost missing completely in Europeans. And once you look at the genomes, you start finding many of these. So none of them are in HapMap. These are 8 mutations in neighboring genes that are non-synonymous, prevalence at least 10% in the West African genomes that we had, in the 118 West African chromosomes that we had, and prevalence 0 or close to 0 in Europe. So now we could go and and the gene, we had 8, we are going to genotype all of them, but the gene, the answer is going to be in this gene ApoL1, which is actually the next gene over. This is MYH9, this is ApoL1, so it's actually the next gene over from MYH9. And 2 of these, of the 8 we found, were in this next gene over. And in fact, it's not 2 but 1, because they are in perfect LD. They are perfectly co-inherited. In that data, they were in perfect LD. In bigger data sets, they are in almost perfect LD. So it's like one haplotype. It's only about 20 kilobase from the best MYH9. So the best MYH9 mutation is not one mutation, but several mutations. But they're actually on the side close to the ApoL1 gene, and they are amino acid changing. So this is very exciting. Now we can do the statistics. So now our small preppy samples that includes like 100 cases and a bunch of controls. We can do the statistics and see that the strongest association signal we find by far are in these ApoL1. So we see that they are not in perfect LD, they are almost perfect LD, but the strongest, the smallest p-values, the strongest signal we find is on these ApoL1 mutations. This is compared to the MYH9 best mutations that we have. Okay, and we can also do an exercise like put this in a model and then see that these are no longer significant, right? So see that this signal is actually explained by this signal. So we have strong statistical evidence that ApoL1 is in fact the one responsible. But does ApoL1 make sense biologically? And the answer is yes, because there was already knowledge in the literature that ApoL1 is associated or related to trypanosomiasis, which is African sleeping sickness. Okay, so African sleeping sickness is traditionally or evolutionarily a big problem in Africa. and humans are naturally immune or developed immune against most of the parasite species. So once you get infected, if you're not immune, you basically die from this not very nice disease. So this is a parasite, and the human serum is able to kill most of the species of this parasite, but 2 species are still responsible for disease in human. And in 2009, they showed that this protein, APOL1, if you change it in some ways, it's able to also kill these 2 species. That basically gives protection. You can give the body protection against these additional species by changes in APOL1. So this was supposed to show that West Africa is actually traditionally and still to this day a place where African sleeping sickness is a major concern. This is the origin of most of the genetic material of African Americans. So one thing we did is test these study these mutations and figure out that they are related to the ability of the APOL1 to kill this parasite. So basically, some prediction mechanism says that they are related to the change in function that allows to kill the parasite. So now we are in mid-May 2010. We are the provincial group in Technion and Tel Aviv University in Israel. We've been disconnected from the NIH people who don't return calls anymore. And what we have is we have something that we believe is a major finding. We have a theory about an evolutionary connection to African sleeping sickness. We have very lousy data on the statistical side, and we have 0 data on the biological connection to the African sleeping sickness. And a very strong sense that we are being threatened by these large, well-funded and data-rich groups who are probably in the same place or ahead of us. So what could we do? We could look for more data, more collaborators. We could try to find a way of doing the biology in the lab, or just try to publish what we have. So we decided to try to publish what we had, and ambitiously sent it to Nature Genetics. And they, as expected, replied, this is very nice, but it's not really solid. We need better data, and especially we need something about the biology if you want this to be Nature Genetics worthy. So we contacted another well-known guy, trying to get more data and other things from him, and he said explicitly that he was part of a group that is making major progress on this general story, and he cannot work with it. Okay, so the conclusion, and then there was one more thing that I wanted to show here, but the video is no longer online. There was a 1-hour talk by this guy, Jeff Koch from the NIH, about MYH9. At Wake Forest, he was giving a highly attended talk about MYH9, and the whole 1-hour talk was about MYH9. This is late May 2010. And in the answering question part, people asked him, what about connection to disease? And he said, well, we should look at different diseases. And he said the word trypanosomiasis, which is African sleeping sickness. And at that point, my doctoral collaborators from the Technion, who I was sure generally always think they are being chased by it, said if he says this word, for sure they know it's APOL1, and for sure they are going to publish it soon. So because of this word, we have to publish immediately, no matter what. Okay, so that was the conclusion from this one, where the mounting evidence was that we are paranoids who are being chased. So, okay, so actually this is a very important slide. So this is our paper, and there are several things we can see here. One, where it was published, this pretty obscure journal, Human Genetics. But what's the benefit of working with an obscure journal and publishing something important in an obscure journal? We submitted the paper on 7 June, and it appeared online on 16 July. Okay, so the point was, we're going to give you this paper, but you have to publish it basically immediately. Review it within 2 weeks and publish it within a month. And now the question is, were we really being chased? And the answer is yes. So our paper appeared online on 16 July. This paper appeared online on 15 July in Science. And it says association of trypanolytic A2L1 variants with kidney disease in African Americans. And when we look in the author list, we find some nice surprises, such as our friends Winkler and Kopp and our other friend Barry Friedman. And we also see that this paper was originally submitted on 4 March 2010. In April 2010, there was a big conference in the NIH about the other gene, led by these people from the NIH, who are co-author on the paper submitted in March, saying that it's actually a different gene that's responsible for it. Right? So they already had submitted a paper saying it's a different gene, and they had a conference on the wrong gene. And this is part also of the cultural phenomenon or big problem maybe in the science in general, medicine in particular, that before something is published, it's not considered known. Right? So even if you know something, you hide this knowledge and you have a big conference and you go to Wake Forest and you give talks about something you already know to be wrong because you haven't yet published that it's wrong. Okay? So, so one question we might ask ourselves, except the gossip and the doubts that we have about the scientific method, was there the justification that this paper appeared in Science and our paper appeared in a third-rate journal, and actually there was. So they were able to actually solve all these things that we would have wanted to solve but couldn't because of our data limitations. Turns out also some intellectual limitations, and we did not have labs to see what these things do to the trypanosomiasis parasite. So one very nice thing that they did is they found an additional mutation, which is a deletion, which we would have been able to find if we knew how to look for it in 1,000 genome. More rare mutation. But when you combine this other mutation with the mutation we also found, you actually get pure recessive mode of inheritance. We found an additive mode of inheritance because we didn't have both mutations. When you include both of them, so you need a copy of one of them, of at least one of them on each of the chromosomes, and then you have the increased risk, and it's purely recessive. I'll show you the proof of that in a minute. And the other thing they did, which is amazing, they actually proved in the lab that this mutation, these two mutations, give protection against that. So the theory we had as a theory, they proved it in the lab. 5 minutes? Okay. So this is, for example, proof of this is proof of the recessive mode of the inheritance. This is these go up top. There are the mutations we found, and we see very strong signal just doing a simple test of association. So this is low 10 p-value. We see weak. Once we remove those, we see a remaining weak signal up top for the 6 base pair deletion. And once we remove both of them, so we include them as explanatory variables in our logistic regression, we see no signal left for MYH9 mutations or any other mutations. Okay? So we've proved that by using these two mutations, in particular in a recessive mode of inheritance model, we explain all the signal, all the association signal in the region. And this is the proof that that these things kill parasites. This is the nicest thing to look at. So actually, one nice thing is that they kill parasites in a dominant mode, inheritance mode. So you need just one copy to kill the parasite. You need 2 copies to get sick. And this is what happens. This is a happy parasite, and these 2 are dead parasites where the thing connected to them and killed them. Okay, so these are actually pictures they were able to generate in the lab, and these are graphs showing that not all of them kill with the same effectiveness, but generally demonstrating the ability of the variants to kill the parasite. Okay, so I think we understand what the emerging theory is, that these mutations appeared in West Africa because they were protective against trypanosomiasis. Some variants, they went through this selective sweep. And at that point in time in Africa, probably kidney disease was less of a major threat to survival than African sleeping sickness, so they were actually positively selected in West Africa. And obviously this moved with the gene flow from Africa to the US. In the US, these African Americans were not exposed to trypanosomiasis. On the other hand, they have increased lifespan and maybe some environmental factors that like HIV, which is not exactly an environmental factor, and then the kidney disease risk of this emerged as a major issue. This is just demonstrating the selective sweep. I don't think it's clear. So actually, people have been working. So this is, we're talking the second part of, or middle of 2010. Since then, many people have been working on the really there's one major piece of the puzzle that remains. What's the mechanism of getting kidney disease with this? So what's the actual connection of APOL1 to kidney disease? We know the connection to trypanosomiasis, but what's the connection to kidney disease? This slide is out of date, but when I try to look today, I don't think they have made any major progress on understanding the biology, the connection between the gene in particular and these non-synonymous mutations this gene in general and the non-synonymous mutations in particular to kidney disease. But there's ways they are using it. For example, they are screening potential kidney donors for these mutations, and they do not take kidney donations from people with the risk genotype. This is one way this is actually medically used. Okay, thanks.